Hello, I'm Professor Khalid Khan from University of Granada in Spain. We are going to talk about evaluation of an infertility treatment, and the trial we are going to discuss is called the Time-Lapse Imaging Trial, or the TILT study. The chief investigator of the TILT study is Dr. Priya, who is with me. Priya, could you please introduce yourself?
Hi, I'm Dr. Priya Bhide. I am a researcher and also a fertility specialist. I work at Queen Mary University of London in the United Kingdom and also do my clinical practice at Homerton Hospital, which is also in London.
Okay, Priya, thank you for that introduction. Uh, the trial we are going to discuss today, Dr. Priya will explain briefly in just a second, but I just want to draw your attention to my two books, one concerning clinical trials and the other concerning evidence synthesis. The key messages that exist in the book concerning how best to deploy these forms of evidence, uh, well, you're going to see that in a true example through the work undertaken by Dr. Priya in the leadership of Dr. Priya. Priya, could you say a few words about the publication?
Yes, so today we are going to discuss the time-lapse imaging trial, or TILT. And this is a multi-center randomized control trial, which is the best kind of evidence to answer any research uncertainty. And this trial assessed the clinical effectiveness and safety of an intervention, which is the time-lapse imaging incubators, which are used for embryo incubation and also for embryo selection in the context of IVF treatment.
All right. So, to set the scene, could you say something about what we are dealing with here?
So as I've said, this is a fertility trial. And since IVF came into practice in 1978, there has been a massive increase in the demand for IVF globally, and more cycles are being performed every year. Now, this increase in the demand is unfortunately not matched by a similar increase in the success rates, which are suboptimal, and they have remained static in the last decade at about 26% live birth per embryo transferred. Now, IVF is an expensive treatment, and therefore, this suboptimal success rate is even more significant from a health economic perspective. So every patient and every healthcare professional who is involved in IVF is keen to improve the success rate of IVF, and hence we are trialing different interventions which might improve the success rate of IVF. And this is where these time-lapse imaging incubators come into the picture. Now, the time-lapse imaging incubators are specialized incubators in the sense that they have a digital camera which is placed inside the incubator, and that digital camera takes a picture of the developing embryo every 5 to 15 minutes to create a time-lapse sequence of the developing embryo.
Priya,
Now,
Priya, sorry to interrupt you. For those who don't know this technology, is this camera very similar to the time-lapse imaging that exists in the mobile phones or handheld devices of that kind?
So the time-lapse, the digital camera was assessed for safety, and it is similar. It takes a picture of the embryo, but the amount of light exposure and the amount of exposure to the embryos was assessed, and this is considered to be a very safe digital imaging, which will not harm the embryo in any way. So yes, very similar.
All right. Thank you for explaining that.
So the images that are created from this can be seen on an external computer screen without actually having to open the incubator and take the embryo out. So this system may be able to improve the success rate of IVF in two ways. One is that it allows the embryos to be monitored remotely without taking them out, and that gives them an undisturbed culture. And this may make better embryos and subsequently improve the pregnancy rate. The second thing that these incubators do is they create a time-lapse sequence of embryo development, which may give more information than is previously obtained with standard technology. And this may be able to help to select the better embryo. Now, if we select a better embryo, then we are more likely to have a baby. So there are two ways in which this embryo incubation system may actually help. Now, when we decided to do this trial, we looked at what work has already been done. And we found that the evidence that was existing at the time was not only low quality, but it was also inconclusive. We spoke to a lot of our colleagues, and there was equipoise. People were not sure whether this actually helps or not. And people were very willing to recruit to a trial if one was started. And that is how we thought and started doing this time-lapse imaging trial. Now, this was a multi-center trial, and we performed it across 7 centers, 6 of which were in the United Kingdom, and we had a very good collaboration with our team in Hong Kong. So we had a central research team which was based at Queen Mary University, as you can see in the pictures. And then we had our 7 recruiting centers from where our patients were recruited. So this was a fantastic collaboration between a lot of centers that performed IVF.
Okay, Priya, just to clarify, at the time of conducting the trial, the technology had already been bought or purchased by several IVF clinics in the hope that they are offering more modern treatment to women. So, certainly, women's and couples had been hoping that with this technology they would get a better success rate, isn't it?
Yes. So, in the context of IVF, I think what we are dealing with is 2 issues here. One is that it is a very rapidly evolving branch of medicine. So, there is a lot of new technology which is coming up. And the second important thing is that the population that we are dealing with is very, very emotive. Patients would be able or willing to go to any length to increase the success rates of treatment. And these 2 things together, put together, is to say that if there is a new technology and it is available in the market, then they are often introduced without robust evidence. Because a patient would say, I'm willing to try it, whatever the price, whether or not there is sufficient evidence, I just want to improve my success rate. So this is what drives the use of this technology without adequate evidence.
Okay, so this technology, one might say, uh, has been introduced without necessarily knowing that it works. There is, in fact, a possibility that it may have no impact on the pregnancy rate that is achieved by people who receive this form of treatment as part of IVF, isn't it?
Yeah, that's absolutely right, Khalid. So this
Okay.
is where the role of a researcher starts. So this is precisely what I said, that we assessed the evidence before we started the trial. And although there were a few studies that had been conducted, the quality of that research was not very good, which means that we were not able to derive definitive conclusions. And we were still very inconclusive whether this actually makes a difference or not.
Sure. Also, in fact, if clinicians were not uncertain, they would not have taken part in a study that randomizes people to not receiving this new technology. So that's another important feature to consider in the planning or design of
Yeah.
a new clinical study. We move on from here. The study got ethics approval. Uh, it, what was the year when you presented the study for funding and approval, Priya? Can you say a few words and describe the study as well?
Yeah. So it has been a long journey to get to the point where this study was completed. We started thinking about the research question way back in 2015. We were given granted funding in 2017. It took about a year to get the study set up because we wanted to be very particular about the way we designed the study. And what I want to stress here is that the quality of evidence that we generate is very dependent on the robust methodology of any study. So we were very, very keen to have a good study protocol and to go through the governance and ethics. So we designed the study, we wrote the trial protocol, we went through all the governance and regulatory requirements for the study. We wrote the protocol with the input of multidisciplinary teams, which were the clinicians, also the embryologists who actually conduct the intervention in the laboratories. We also included patient and lay public representatives because it is important to work with them. And of course, we had very experienced trialists. So we wrote the protocol and we published it before we completed recruitment. The trial was also registered prospectively on the International Trials Registry, which is now a mandatory requirement.
So,
So what
Priya,
I
may
can,
I just ask you a clarifier? The trial in its designed form was available on the trial registry from 2018 onwards, and subsequently, the full protocol of the trial was also available publicly. Did anybody make any comment about your trial or our trial design at any time?
uh, I think it was quite well received because people were quite, clinicians working in the field were quite keen to get an answer to this question. And we were actually encouraged and applauded for having this ambitious project and doing this study.
All right, thank you. Well, let's return to the study design. What were the comparisons?
So, as I have said before, this intervention may work in two ways. One is the undisturbed culture, and the other is the additional information, which is called morphokinetic parameters. So, because there are two variables designed in the system, we had a unique trial design and we designed a three-arm trial because we wanted to know what is the overall contribution of the time-lapse imaging system and also the independent contribution of these two variables. So we had a three-arm study and we were able to get the answers for all these questions in a single trial. So our first arm was where we
Apologies.
First arm was where we randomized participants where they would have the entire time-lapse imaging system, which had the undisturbed culture and the morphokinetic parameters. The second arm gave the embryos only undisturbed culture, and we selected embryos using standard methodology. And then, of course, we had to have a control arm where we had standard incubation and we also had standard selection. Now, we wanted this trial to relate to real life scenarios. So we wanted it to be a pragmatic trial. We allowed the centers to use the IVF protocols of their choice. We allowed the centers to use the time-lapse machines of their choice. We
Okay.
kept the inclusion criteria to be very broad
Mhm.
so that we could include as many participants who were undergoing IVF to take part in this trial. And generalizability is important because it relates to real life scenarios in everyday practice.
Okay, thank you, Priya, for explaining that. Let's move on. How big was the study?
So uh, we thought it was important to have a primary outcome, which
Mhm.
is important to all stakeholders, which is the patients, the healthcare professionals, policymakers, and funders. And for fertility trials, there is only one outcome which is important to everyone, and that is live birth.
Yeah.
So ours was the only and first study which was adequately powered to detect an increase in live birth rate. And we had an effect size of 8.75%. And that calculated to 525 participants in each arm, giving a total of 1,575 participants, which is quite a big number.
Well,
So it was a large study.
yes, not only it is large, its size is determined according to a scientific assessment. How did the study go from this point onwards?
So we started recruitment in 2018. And we consented more than 2,000 participants to be able to reach the pre-specified sample size of 1,575 participants. The trial protocol was designed well, and because of specific time points in the trial protocol when we would randomize participants, we had zero withdrawals after randomization, and that was a sign of a well-conducted trial. I also would like to stress that we had outcome data available for close to 99% of the participants. And again, that is very important. We had a very minimal crossover or allocation non-adherence, which means that we had an identical population for a per protocol and ITT analysis. And irrespective of the results, what this says is that the data generated by the STILT trial was very, very high quality.
Okay, thank you. Let's cut the chase. Where can we see the main findings? What are they?
So our primary outcome was live birth, and we had two primary comparisons. One was to assess the effect of the system overall as compared to standard, and the second was to assess the impact of undisturbed culture compared to control. There are a lot of confounding variables in the IVF scenario. So we used what is called a logistic regression model, to which we added all these confounding variables. And we presented the results as something called an adjusted odds ratio. And we were unable to find any significant difference between the groups for both the comparisons for live birth, as you can see in this slide.
Uh, Priya, excuse me if I missed it earlier. I think the statistical analysis plan as to how data will be handled was finalized before the data set was opened and released for analysis. Is that not correct?
Absolutely, yes. I'm sorry I missed saying that.
No.
A very detailed statistical analysis plan was put together by a statistical team, and it is important that we pre-specify the analysis that we are going to do. And we finally sign off this statistical analysis plan before the data is locked for analysis.
Okay.
So this is again an important factor which tells us that this trial was a well-conducted, robust trial. Yes.
Okay. So although study wasn't powered for, is never powered, usually never powered for secondary outcomes, did we observe any other hint of effect in secondary outcomes?
We assessed the trial for several secondary outcomes for effectiveness and for safety. So outcomes such as clinical pregnancy rate, miscarriage rate, or the rate of twins. And we were unable to find any significant differences for any secondary outcome for either of the comparisons.
All right. So it looks like that based on what you found, the promise made by the technology has not shown itself to bear effect in terms of better pregnancy rates for infertile couples.
Yes, so there is biological plausibility for a lot of interventions, but this biological plausibility doesn't always translate to improve clinical outcomes. And for us to know if the clinical outcomes are improved, one must run a good trial like we have for us to be able to know if we can use this intervention.
Okay, so let's just look at the post publication scenario. The Lancet uh, published the trial. Obviously, at that time, the results all became public. Then I understand that some people who use or like or have an interest in the technology have not liked your results. So, and they obviously have the right to comment and express their opinion. But it's important to disengage factual information from opinion. So when a commentary had been written about this trial um, in RBM Online Journal, Priya, we had to reply and more or less emphasize that this study had been carried out according to a pre-specified protocol, which is pre-registered, which has a priori statistical analysis plan. Therefore, as far as the results are concerned, they hold. Whether you like them or not is a matter of opinion. It is not a matter of fact. So we've taken this study all the way from its conception through to its approval and funding, and then to completion and publication, and then also through the post publication comment or peer review phase. What might you say, Priya, are the conclusions that can be drawn?
So this trial confirms and gives definitive quality, high quality evidence. And it tells us that there is no benefit for the use of these time-lapse imaging systems to improve the live birth rate in women undergoing IVF or ICSI. So the offer of this intervention to women with the expectation that it might improve their pregnancy rate or their live birth rate cannot be justified. And although this is a negative trial, that it shows no difference in outcomes, I think this is really important information for all the stakeholders, more importantly, patients, also healthcare professionals who may think that this technology actually works, to funders, especially in the context of where this is a publicly funded medical treatment, because the technology is expensive, and also to policymakers. So I think this is really important information for all stakeholders involved in the IVF process.
What is your plan for future research, Priya?
So this trial is powered to live birth. It tells us that in the pragmatic setting, the intervention does not improve live birth rate. But there is some speculation as to whether it may be useful for some subgroups or it may be useful for some important secondary outcomes such as miscarriage. Now, this trial is not powered to detect such differences, and nor is any other trial that has been conducted so far. So if we do want to know the definitive answers to these questions relating to certain patient subgroups or to some specific secondary outcomes, then we need a larger data set. It is very, very difficult to have such a large trial to detect these outcomes in subgroups for secondary outcomes. The better way is to pool data from already completed RCTs. And this is something that we call individual participant data meta-analysis, where we pool the data from several randomized controlled trials to gain adequate power to answer these questions. So we are currently in the process of doing an IPD meta-analysis, and we have data sets from several well-conducted RCTs. And I hope that I might be able to present this to you in the near future.
All right, Priya, thank you very much.
Thank you so much.